My name's Liz Patton. I'm a program leader at the MRC Human Genetics Unit and a professor at the University of Edinburgh.
Professor Patton, could you tell us more about the research focus of your lab and the current question that you're currently investigating?
Yeah, sure. So we're really interested in understanding the biology of melanoma and identifying small molecules or drug leads to target melanoma. And in particular, we use the Zebrafish system because we can look at the developmental biology of the melanocyte lineage. So melanoma arises from melanocytes. So we can study the natural biology of the melanocyte in an animal and then follow how that melanocyte lineage, how the development of the melanocyte goes wrong in the progression to melanoma. And then in particular, because we can use these two stages of the zebrafish. So one is an embryo, they're very small, we can do lots of live imaging, and we have the adult that we can develop these melanomas. This really allows us to screen for small molecules and drugs in the embryonic stages and then try to see if we can target melanoma in the adult. So that's sort of our big question is, can we identify new targets in melanoma, particularly thinking about the developmental lineage that goes wrong in melanoma, and can we develop new drug leads?
That's really fascinating. But so you try to identify targets from zebrafish model and then try it for the validation in the human studies, which is very fascinating. Could you for a second, because I have a very limited knowledge on zebrafish model. So what is the reason behind choosing zebrafish as a model organism to study melanoma?
So one reason is because of this power of being able to use the embryonic system. They're really small, they're like caviar, they're microscopic. We can see melanocytes developing in their natural context. So it's very, very powerful. We can see what goes wrong when we start to genetically modify those fish with genes that are mutated in melanoma. So that's one reason, is the power of being able to use that developmental side. The other reason is, as an adult, they develop melanomas. These are true cancers. They are proper melanomas. They have similarity to some of the melanoma subtypes in humans. And in fact, even though zebrafish may feel very different than a person, about 70% of the genes are highly similar between zebrafish and humans, and over 80% of disease genes are highly similar. So in fact, they're a fantastic disease model. They kind of fit in this space between the model organisms like yeast and C. elegans, which allow you to do large-scale, big genomic questions, and maybe a mouse or more mammalian model, which require you to think about fewer numbers, more sort of targeted questions. So they kind of fit in this intermediate space. They're vertebrates, so they have a lot of similarity to us, but they're small, they're born outside, or they're fertilized outside of the mother, so we can collect hundreds or even thousands of embryos quite easily from the fish. So they sit really nicely in this scientific animal model space.
Wow, that is really revolutionary approach as well, and equally fascinating at the same time. So what's your research vision for the next 5 years?
Yeah, so the thing that we're really interested in now, so I sort of spoke a little bit about the development of the melanocyte lineage. That is true, those pathways go wrong in melanoma, but it's really clear that not all the cells in the melanoma look the same. There's heterogeneity. So even though you have a tumor that's growing, invading tissues, it's eventually going to kill the fish. If you start to look at the individual cells of those melanoma, you find actually they have a lot of differences between them. Some are at the genetic level, but there's a lot that are at the gene expression level. And we're really interested in this this small population of cells that express an enzyme called ALDH. We don't really understand the function of what that ALDH enzyme is doing in the tumor, why it's so important, but it seems to have those cell populations seem to have important properties. They seem to be more able to start tumors than other cells. They seem to be resistant to some types of drugs. They seem to be resistant to other genetic changes. So we call it sort of phenotypic differences within that tumor. And there's also a lot of plasticity between the cell types. So you think, oh, now I understand this cell type, but actually it can change and become another cell type. So the big vision for me would be that you could have a zebrafish where we can mark all these different populations and follow as these cells change in their heterogeneity, and then see how that is altered with drugs and with genetics.
Although I have very limited knowledge with zebrafish, but the way you just described, it's so fascinating. It seems like you're very passionate about your research.
They're fantastic animals. You never get tired of them. It's always a pleasure. You know, when you come in, look under the microscope, it's always exciting.
That's great. So how did you get motivated to do this career or choose this career of research?
Yeah, so I did a really wonderful undergraduate project in the university, Dalhousie University on the east coast of Canada. And that was in Jerry Johnson's lab. And he had worked with Lee Hartwell, who won the Nobel Prize for identifying cell cycle mutants and cell cycle genes. And so he really introduced me to the concept that you can do genetics to understand these fundamental mechanisms of how a cell divides. But he was also, even though we were just using yeast, this is the same yeast that you use to bake bread and to make beer, he really introduced me to the concept that what we were learning was going to be important for cancer. And he was very passionate in his outreach, in particular with the Terry Fox Foundation, raising money for cancer patients, even though we were just working on yeast. And so then I moved to Toronto and worked with Mike Tyers, again working on yeast, again trying to understand basic cell division processes using genetics, using genetic screens. And we had a very productive time. Again, always thinking that what we were learning at the fundamental level could be important for cancer. And by the time I was finishing my PhD with Mike, I really was kind of interested in cancer. So I loved what I was doing, but I was always kind of explaining why it was relevant for cancer, and I was ready to actually use an organism, a model system that allowed us to look at proper cancer. And so at the time, Len Zahn at Harvard was also interested in using zebrafish to study cancer, and in particular to do cell cycle screens. So I went there to study with him and to work with him. And we did genetic screens looking to understand cell cycle genes. And then there was a paper published, seminal, fundamental paper in our field, that BRAF was mutated in melanoma.
Is it 2003?
Yes. And so he just said, I think as a side project, we should be doing this. And worked also with David Fisher at the time. So we got the BRAF gene. I was told by people, you'll never get a transgenic to work in zebrafish, this will never work, and it really did. And it was very, very exciting. And so what that showed for the first time in an animal model is that BRAF could drive a nevus formation, and then an additional mutation, in this case P53, allowed that nevus to become melanoma. And so now lots of people use that model and are really making very exciting discoveries. So that's how I got kind of into cancer. It comes from a very basic science.
You mentioned about hard times, so I'm going to shift some gears now, asking about, so your career seemed like you traveled a lot and did some of the fantastic work while you were at early stages. Now you're doing some truly revolutionary work when you're a PI. So what were some of the major challenges that you had faced in your career?
Yeah, so I think I should say that undergraduate, PhD, postdoc, fantastic mentors. So I think the challenges that I faced at those stages were like what we were just saying. Oh, this doesn't work, it's so hard. You have a great idea, it doesn't work out, something technically is beyond what you can do. those kind of challenges you face a lot. So you need a lot of resilience. Friends help you through that for sure, you know, forming friendships in the lab. One of the challenges that I would face, maybe sort of at a bigger level, is I moved from Canada to the US, which maybe isn't such an unusual move, but it's a big move for me, you know, as a person.
It is a big move, I would
yeah,
say. You're moving across the country.
yeah. So you sort of move, you know, you step out of your comfort zone a little
Different
bit.
culture.
Different culture, even though they are similar cultures, they're still different.
Yes.
You have to adjust to new cultures, which is wonderful and one of the amazing things about science, but it's still kind of tough. You know, moving to a new place, figuring out a new system, stepping away from a group of people who would have supported you if you stayed. You know, so that's quite tough. And then you have to make new connections yourself, meet new people. Incredibly rewarding. I feel very well connected both in the US and in Europe, and I'm really love those interactions. They culturally think differently about science. Their approach is often very different, very complementary but different. So I love being in that position, but it doesn't mean it's easy every day.
Yeah.
So I think that's been one of the challenges, adjusting to different cultures, figuring out the system, because even though of course science is about the science, it sits within a structure, you know, a system. And so figuring that out is always a challenge.
But would you say that if you had a good mentor, then majority of the challenges can be taken care of? Apart, like in terms of your research?
Yeah.
I understand that a mentor can't really do anything when your things are not working, but I suppose you can get some good advice in terms of switching the projects, maybe because not every project will succeed, and that you have to be smart enough to make a decision.
I would agree with you. I mean, good mentors are wonderful, essential, but ultimately it's you. You know, are you going to make the decision to own this work or not? And you become the expert. So you can get advice, but it's ultimately your project and your life. And you know, I've had lots of great help along the way, there's no question, and even at my stage now I get lots of help from friends, mentors, colleagues. When I'm writing a grant, when I'm preparing to give a talk, I'll call a friend in another country and practice by Skype. You know, I get lots and lots of help. But there's also another side, which is, and I would say I'm guilty of this, is caring too much about what other people think, taking too much advice, and that can hold you back, because ultimately you're in the driver's seat, and if you have too much advice, you can't hear your own words, you can't hear your own thoughts, and ultimately that's the special contribution you're trying to make.
Yes, I see. Yes.
You can be over mentored.
Yes. Over mentored is the term. So trainees always have to ensure that they are on the driving seat.
Yeah, I think so. I mean, definitely when you're in a trainee position in a lab, you know, maybe there's some different dynamics there. But I would say, yes, I mean, ultimately the trainee needs to own the work, you know, and that, of course, I'm there to support, shape, mentor.
Yes.
But ultimately it needs to come from you.
Yes. In fact, it is our career, so it would make total sense, because your mentor is just trying to shape you.
And there's no point in, well, I was going to say, I didn't quite. What's so special is having a group of people who are thinking as scientists. So they might be at different stages of their scientific development, but ultimately I want to interact with you as a scientist, not a technician. If I needed a technician, I can hire a technician. I can hire robots. I mean, technicians often can be excellent scientists also, but in thinking about the scientist, ultimately that's the interaction you want to have. And I'll never forget my PhD supervisor saying to me, when I joined the lab only recently joined the lab, and he said, I really don't want to work with students, I want to work with scientists. And I was the student, and I thought, I got it. I hear your words. I will take it up a step.
I would have been like, whoa,
I will take it up a notch. I remember thinking, got it.
That's good.
I know what you want from me.
I think that's a very good message.
Yeah.
That because in the end, PhDs are getting trained to think scientifically, and it's a scientist in making. So you should really embrace the scientific thinking skills, and rather than just.
And I think being able to think scientifically is a very important skill. So that's something that we have, created in human culture, and it's not something that comes naturally. It's not. I know it feels like in our world that we live in that the scientific method, people would just apply that to everything. But they don't. It's something we have to learn, and it has real value. It asks for evidence, and you can use that not just in a scientific context, but in your approach to the world. Is there evidence for this policy? What happens when we do this policy? What happens with our control group? So before you take an idea and apply it to a whole group of people, what's the evidence? Rather than people's gut feelings. And so I really passionately believe, even for PhD students or postdocs who choose not to pursue a scientific career, that their training is so good. It's so special and important for human culture, for our society, to be able to have a scientific approach.
Definitely. So in your opinion, what do you think about representation of women in science, especially in STEM?
So in my direct experience in Scotland, and I know it's different in different countries, we have lots of PhD students who are female. So definitely women are interested and studying science. We have lots of women as postdocs. So lots of women who are clearly talented, able, and are passionate about their work. Then there becomes this transition to the PI, and we seem to lose a lot of women at this stage. And I think there's a lot of reasons for that, but that's clearly a bottleneck time. Then it seems that when I look around where I'm from, anyhow, there are quite a few women PIs, but many of them kind of stay at a certain level. They don't then grow into the leadership positions. So these, as you say, they're debated. Why does this happen? What are the factors that are holding people back? What are the unintended biases? What are the intended biases? So I think we need to find that out. It's definitely not. It's definitely not a lack of talent. We have a talent pool.
Yes, as you said.
Yeah.
So how would you promote women to sort of be more at the administrative level? I understand we do need to explore the bias, and I guess if we found that there is a reason that is why so, then we could potentially overcome that, and having overcome that, we could have a society where women and men are equally at equal positions.
Yeah.
But irrespective, given the current scenario, how would you promote women?
What can we do? Yeah. So I think there's some. I think there are sort of different forces that come into these things, and I should say I don't think it's just women. I think there are probably other groups, minority groups that have the same. I don't really know those issues, but they're clearly very important. I mean, we have very few minorities in our institute, that's for sure. And we have very few even at the PhD student level, so I think there's another problem. So I kind of course I can't give a fully comprehensive answer, but I could maybe talk about a few things that are on my mind. So one of them is a very practical thing, and that is that at that transition zone from postdoc to PI, a lot of women want to have their families. So there's sort of there's a practical issue that arises. It's also the same time when you have to produce at this super high competitive level. And so that's not always compatible with having a young family. And I think I see this also with men who are involved in their families. So clearly having young children can be quite tough at that time. And I think that tends to fall onto the women, in part from the biology, but also maybe there's expectations that they'll do more or caring for the children. And also you want to care for the children. I have 2 children. I love spending time with them. Definitely your heart can hurt, you know. So I think there's some practical issues. And I know what helped me a huge amount for both children I had was on-site daycare. And that just made everything work. So where I did my postdoc, they had a daycare there, an excellent daycare. It was the Children's Hospital daycare. The daycare providers were wonderful, warm. I felt completely comfortable leaving my daughter there. And it was the daycare that they used to train the young doctors because the young doctors worked with so many children who were unwell, and a lot of the young doctors didn't have children yet. They wanted the young doctors to see what normal behavior was like. So they would come in and, you know, see the children, spend time with them. So you felt like, oh, we're part of a group together. And then the other thing that did was I had really good friends in the lab. And, you know, we talk sometimes in science about bonding over like going out for drinks or something like that. So we bonded at the nursery and at the playground. So then what you find is that the people in your lab, we all have something in common. We all have 2-year-olds. We all go to the playground on the weekend. We talk science. We help each other with experiments. We pick each other's kids up. So like that's all wonderful. That like helps you a lot. And then when I moved to Edinburgh, my son went to the University of Edinburgh daycare. That was close to my husband's work. And they, for the psychology students, would do psychology experiments with them. So like you felt like it's part of the university, you know, it's just like a wonderful, really, really wonderful. So that, I would say, was a huge help to us. Because the other thing that's happening during that time is you're moving often. That's one of the great things about science, one of the challenges about science. But it also means you don't have your mother and father maybe right there to pick a child up at 3, you know. So you have these pressures on you. So I think that's one easy thing. You know, I think you can have on-site daycare, build networks, support young scientists coming up together. You know, when you have small children, you can also feel very isolated, and you can sort of become disconnected from a group. So I think when you have on-site daycare, you find these commonalities with your peers. So I think that's really, really important. On the other side, where are the women facing these barriers? Definitely some of it are biases, coming against you. You having to demonstrate more. You have to get more grants than somebody else, more papers. You have to have more to get into a top-tier journal than other people. And so that's very, very hard. You're sort of trying to constantly prove yourself. The expectation from other people about how, you're a woman, you should behave. I want to go work in your lab because you're a woman. Well, what does that really mean? Like, what are you looking for from me? Because you might be disappointed. I don't really know what being a woman is, so you might be disappointed because I don't fulfill your criteria of womanhood.
Yeah, to be honest.
Well, it is, and you don't hear that the other way. People don't say, I'm going to his lab because he's a man.
If you just have a daycare, for example, you came to this conference and there are daycare facilities, then your life just becomes so easier.
Absolutely, and I think this one does offer.
Wow.
Yeah, and I think it's also good for men. For example, my husband has been very involved in raising our children, and it did hold him back a bit at times, on the career progression. But, in his department there are not many people with children, and in his department they're mostly men. So they maybe don't understand the same way. But I think it's good for men and women to meet each other in that space.
Definitely.
Because ultimately, at the end of the day, having kids is great. It's great.
I don't know that yet, so I can't
it's
agree,
great.
But
it's wonderful. Yeah.
I look forward to feeling that feeling.
Yeah.
So the final question is, you have been coming to the SMR meeting for a couple of years now.
Yeah.
What do you enjoy the most about this meeting, and how is this meeting different from the other society meetings?
So this meeting's focused on melanoma. So I go to a lot of conferences. I go to many on zebrafish, and I go to pigment cell biology and melanoma meetings. But what's special about this one is it's all melanoma, unapologetically. So, and it takes you right from the basic biology of melanoma through to clinical trials. So to have that all in one space is really, really important.